Hi everyone, and welcome to my second lecture on capillary fluid exchange. Now normally we talk about capillary fluid exchange typically during the cardiovascular lecture, but I actually moved to this point in the semester simply because I think it coincides nicely with the fluid exchanges that we just went over in the first lecture. So a little background, you can see here, this is Marcello Malpighi, and he is actually known for, or is the founder of, the microscopic anatomy. And this was in the 1600s. And so he was actually the first person to identify capillaries in an animal. Up until that point, they weren't sure how arteries connected to veins. And so he was able to discover that, and that's the reason he's there. And it's not just because I like putting surly-looking gentlemen on the cover of my lectures. Now at this point, I'd like to just give you like a brief overview of the vascular system. We will come back to this and touch on it more later on, but the basic layout of the systemic circulation is that you have the heart, which is a muscle and acts as a pump, and it pumps blood into the arterial system. Now in the arterial system, that then branches and continues to branch until finally we have smaller and smaller arteries, which we call the smallest of which the arterioles. Then finally the arterioles, they'll branch into capillaries. And then from the capillaries, we then recoalesce essentially back into the veins. So we start off with the venules, which then become larger and larger veins, which then lead back to the heart. All right, and that's the basic layout, if you will. And so in this case, when we talk about the capillaries in particular, we'll come back to arteries and veins later. But in particular, all right, so with those capillaries, is that the capillaries are the most numerous. They have very thin walls, thinner than the arteries and thinner than the veins. All right, the arteries have the thickest wall and the most muscular, and the veins are smaller than the arteries, still with some musculature, but much less than the arteries. But the capillaries don't have musculature. What they have is just an endothelium, which is the cells lining the inside lumen of the capillaries, as well as the inside lumen of both arteries and veins, as well as a basal lamina. So it's very, very thin, and that actually helps us to make exchanges that are going to occur between the capillary and the tissue. So understand there are lots of capillaries in all the organs, because that's really what we call sort of the business end of the circulatory system. This is where the exchanges are going to occur to bring nutrients, for example, to an organ, or take away wastes from an organ. Okay? And this capillary exchange that occurs with these tissues, it's a two-way movement of fluid. All right? There's going to be what we call filtration, where there's going to be things that are moving out of the capillary. There'll be diffusion of substances out of the capillary, but there's also going to be diffusion of substances into the capillary, because this is a two-way exchange of not only supplying but also removing. Now, as I mentioned, the capillaries themselves, these are the exchange vessels, and I mentioned it's sort of the quote-unquote business end. This is where the exchange actually occurs. This is how the tissues are going to interact with the vasculature and get the nutrients that they need and get rid of the waste that they need to get rid of. And what you can see over in the cartoon over there is that you have on the artery side, you can see how thick the artery is. In fact, the layers, right, there's several layers, but the main ones, let me use green here. All right, so you can see in bold, you have the tunica intima, it's still red there, we go. The tunica intima, the tunica media, and the tunica externa. Tunica just means like a tunic or a coat. All right, that surrounds it. And you can see the lumen in the center, where the red arrows are drawn. All right, that's the lumen of the artery, and you can see the tunica intima has the endothelium, which are the cells that line the inside lining of that lumen there. And there's the subendothelial layer, which is just beneath that, right? And then there's an elastic membrane, and you'll notice in the media is a thicker portion of where we have our smooth muscle. And then just outside that, you have the tunica externa. All right, those are sort of the general things. Again, we're going to come back to a lot of these details later on in the semester. You'll notice that it'll branch, and when it branches, these are really becoming smaller and smaller arteries until we finally have the arterioles, and they become the capillary. Then you have the capillary network there, and then from the capillary, or I should say actually, let me point out this. In the capillary there, you see that the wall is very thin. It's just an endothelium and a basal lamina, and it coalesces back into the veins, and the veins become larger and larger until you have the superior and inferior vena cava returning to the heart. All right, and so you see the veins, they have the same 3 layers, but the layers are much thinner because the musculature of the veins is much less than the arteries. Okay, and that's the basic design, if you will. Okay, so there are many types of capillaries throughout the body. One of the ones that I'll be referring to probably more often is the continuous capillaries, which occur in most tissues. In the continuous capillaries, you have these endothelial cells that form these tight junctions, so they adhere to each other very tightly. And what happens is they have some areas where they're not as tightly bound, and this can actually form like a tube that twists between the endothelium, or like a pore, if you will. And we call this an intercellular cleft, all right, which is essentially, it's a cleft or a gap between 2 cells. And they can vary slightly in sizes from one type of continuous capillary to the next. Some continuous capillaries have more pores than others, okay? But the important thing is that these only allow the passage of very small solutes like glucose or sodium chloride, for example, whereas things that are larger, like proteins, would be excluded. All right, they wouldn't be able to make it into the pore, or cellular components like red blood cells and white blood cells. Okay. Now there is a subtype of continuous capillary, which is known as the blood-brain barrier, as I have in the parentheses there, which let me highlight that for you. Blood-brain barrier, or the BBB, okay, and the retina, for example, where they have a continuous capillary, but there are very, very few intercellular clefts. All right, where it's significantly fewer. All right, and this actually helps to protect and regulate what goes into the brain and towards the eye and so on. So there's a purpose to it. Now, with fenestrated capillaries, fenestra means window. So these are actually cells. The endothelial cells themselves have windows or pores in them, as well as they can have intercellular clefts. And this allows for the passage or the passage of more solutes, more like say in bulk, and it would occur in organs where we would want to see or want to have more exchange occurring, right? So for example, like in the kidney, where we're doing a lot of filtering of various substances, small intestine for absorption, also endocrine organs, which are secreting a lot of hormones. So you might see fenestrated capillaries in those types of organs. Now they require rapid absorption or filtration, so that's why fenestrated is perfect for that. And you can see actually in the cartoon at the bottom here, these are the examples. Here's the fenestrated one. You can see the endothelial cells, which this will be a nucleus of one of the endothelial cells. Here's the nucleus of another one. You can see the junction between them right here. And you see it kind of looks like it's all dotted with holes there. Those are actually the windows, if you will, or the fenestra that are in the cells themselves that are filtering. Whereas over here, this is the continuous, which has intercellular clefts. So between the cells, there's little pores there. There's no holes in the cells themselves. There's no fenestrated pores. The last example I have here is the sinusoids. Sinusoids are also referred to as discontinuous. Okay, these are found in the liver, bone marrow, and spleen. All right, and these are quite large. You see the intercellular gap here is 1,000 times larger, let's say, than the intercellular clefts of the continuous. And this is actually needed because we have to produce, for example, red blood cells and white blood cells, and then deposit them into the blood. So, for example, in the bone marrow, where we produce red blood cells and white blood cells, how they get into the bloodstream is through these large sinusoidal capillaries. All right, so these pores are large enough to allow cells to move in and out of the blood. So this again would allow proteins, would allow anything that's in the plasma because these are very, very large. All right. Again, you would see that in the liver because the liver does a lot of filtering there, as well as the spleen to allow cellular components to move into or out of the vasculature. In terms of capillary exchange, there's really 3 routes where the exchange occurs. There's through the intercellular clefts, which I've mentioned, all right, or those gaps between the cells. The fenestrated pores, which I've also mentioned on the previous slide, but also through what we call penocytosis, right, an endocytosis type mechanism, where we can take in larger substances that may need to cross the capillary and aren't able to do so in, say, a continuous type capillary. and so they'll actually be engulfed by the endothelial cells and move through the cytoplasm and then be distributed on the other side. Now, in terms of the mechanisms in which the capillary exchange occurs, the mechanism is diffusion, and I put diffusion in bold because diffusion is the main way in which exchange occurs. In other words, if I have sodium chloride in the vascular space or in the plasma, it will diffuse down its concentration gradient into the interstitial space. All right, and this is the primary way in which most of the substances are going to move. And that's just one example. Glucose can also go down its concentration gradient from the vasculature into the interstitial space, as well as things from the cells, right? So they may be producing a lot of lactic acid or CO2 from cells that are metabolizing. And so you may have a buildup of CO2 and lactic acid in the interstitial space there. So there's a high concentration there, but low in the plasma of the vasculature. And so it's going to move down its concentration gradient from the interstitial space into the blood plasma. So it can move in both directions. Okay. Again, diffusion really just means it's moving down its concentration gradient. We can also have transcytosis, which again is just moving substances across cells via an actual mechanism that requires ATP. Or we can have filtration and reabsorption, which is really referring to the bulk movement of water. And when water moves, it can also carry solutes with it. And this cartoon is really just to kind of depict that, the various ways in which we can kind of filter, if you will, and diffuse. So you have here, filtration. So if this is, one of the nuclei here, you can see if this is all part of it. This is all part of the wall here. And there's little gaps there, which are the intercellular clefts from other cells. So it's labeled there intercellular cleft. So that would be the space between the cells. All right, we're looking at a cross-sectional view, so it can be a little bit difficult to kind of visualize here, but these would be intercellular clefts, or the spaces between the cells. These up here would represent, the fenestrated pores or filtration pores, all right, which is actually within the endothelial cell itself. Transcytosis is one of the processes which we do exchange, right? We move it across the cytoplasm to the other end of the cell. We can have diffusion, again, down its concentration gradient, simple diffusion. And so, as I mentioned before, and I'm just going to reiterate, diffusion is the major way solutes get to and from the circulation to the cells, or from the cells to the circulation. And another thing I want you to kind of note here is that the capillaries themselves, you notice in the center here, which I haven't mentioned yet, was the red blood cell. It has that discoid shape to it. The red blood cells, okay, they're usually about 7, maybe 10 microns in diameter, so they're very, very tiny. The capillary, the diameter of a capillary is very, very small. In fact, it matches. It's about 7 to 10 microns in diameter, which means that if a red blood cell has to move through a capillary, it essentially goes in through the capillary single file. Okay, because it is too small to accommodate more than one red blood cell going through any particular area at a time. It also means that it could be a tight squeeze, and so red blood cells, it helps that they can be flexible, and when that shape helps them to bend and flex as they move their way through these very tiny spaces here. All right, so here we have diffusion of lipids versus soluble substances. All right, again, kind of remembering that lipids, which are hydrophobic, will tend to move through lipid membranes, like plasma membranes, very readily, whereas water soluble would actually have to take different routes. They wouldn't be able to move through plasma membranes, so therefore they'd have to move through the intercellular clefts or be transported through transcytosis. Okay, so diffusion, right? Glucose, for example, would have to go through one of those intercellular clefts. A lot of the electrolytes, like sodium chloride, would also do the same thing. Now, oxygen being in high concentration in the arteries, would go down its concentration gradient into the tissue because the tissues are utilizing oxygen, so they have a low concentration. And but oxygen would be lipid soluble. It can move through membranes pretty rapidly, all right, but it would diffuse very quickly through a very a simple diffusion type process. CO2 does the same thing. Only it would be doing the reverse of oxygen. It would be going from the tissues into the vasculature. Steroid hormones are another example of lipid-soluble substances that can diffuse and move through membranes readily. And the large particles, however, like proteins, would be, or some of the lipids that are very large might get held back. And in those cases, if we're going to transport them, or there's the need to transport them, then the endothelial cells can do that via transcytosis. And of course, I had to put in parentheses there, except when there's sinusoidal capillaries, because as you recall, those are very large intracellular clefts. And so then red blood cells, white blood cells, proteins can move very freely in those particular capillaries. Okay, so here what I want to depict is just the capillary. And as you can see, I just kind of marked over here in the top left-hand corner. The little red circles I drew actually represent the endothelial cells, and the black line on the outside of the endothelial cells represents the basal membrane. And so we're looking at basically, a section through the capillary wall. So you can imagine that if this end is the arterial end, and over here is the venule side, blood is flowing through the capillary in that direction. Okay. And then out there, the green boxes that you're seeing are actually individual cells. Right, so these are the cells out here, and whatever cells they happen to be, right? They don't represent any particular cells. And then, of course, the fluid surrounding the cells is the interstitial fluid. Okay. Keep in mind that the interstitial fluid and the fluid inside the vascular compartment of the plasma of the blood is all part of the extracellular fluid. Right? And the cells themselves have fluid inside them. We call that the ICF, or the intracellular fluid. Now, in terms of diffusion, we have many molecules and substances that are dissolved in the plasma of the blood. So, for example, I could have sodium and chloride, obviously not combined, but in their ionic states. And the sodium chloride is small enough to actually be able to move through the intracellular clefts. So it would be able to make its way out into that interstitial space. And the way it actually makes its way out primarily is via diffusion. So we say diffusion, really again we're referring to the fact that it's going down its concentration gradient. All right? And in fact it equilibrates and goes down its concentration gradient very rapidly, so that by the time the blood reaches the end of the capillary, the sodium and chloride could have actually equilibrated 80 times over. So this is a very rapid process. So it will come into equilibrium very rapidly from the start of the capillary, and it'll be maintained throughout. Now it is positively charged, okay, and the chloride is negatively charged. And so therefore, since there is a charge, it can't pass directly through the endothelial cells, and so it has to go through those intercellular clefts, okay? But it does freely move from the plasma into the interstitium, or if the concentration is higher in the interstitium, it'll move from the interstitium back into the plasma. And again, it's really just following the concentration gradient here. So that's what's important to understand here, is that it really equilibrates, but it has to go through those intracellular clefts. Another one similar to that would be glucose. Glucose is a small charged molecule. So again, it can't pass through the plasma membranes, but it can diffuse through the intracellular clefts, and that would happen very rapidly. In addition to the diffusion, you will have water moving, and water will have dissolved solutes in it, and so that would be something we refer to as filtration. So if the fluid is leaving the capillary and going into the interstitial space, then you have what we call filtration, and so you'll get some bulk movement of solutes that go along with the water, but that's not the primary way in which these substances are moved across this barrier here. And then, of course, other small molecules, things like amino acids, right, which would also have to take the intracellular path. Now, there are some lipid-soluble substances, and when it comes to lipid-soluble substances, for example, one that we've mentioned in the previous lectures, maybe something like urea, CO2, oxygen. These can actually readily just simply diffuse through plasma membranes. They don't necessarily have to look for the intracellular clefts. Okay, so urea and oxygen would be, let's say, in higher concentrations in the plasma, so it could diffuse out into the interstitial fluid, or if they're in higher concentration in interstitial fluid, it would diffuse in the reverse direction. So again, following just those concentration gradients. So in sort of an example we'll get to more later on, if the arteries are supplying blood to the tissue, they're going to be high in oxygen concentration, and the tissue, which is needing oxygen, is going to be low in concentration, and so there it will diffuse out of the plasma and into the interstitial space and ultimately into the cells. Again, always going down its concentration gradient. Whereas the metabolizing tissue may be producing a lot of CO2. So I'll put some CO2 over here, CO2 out here in the interstitium. So there's a high concentration in the interstitial fluid. And so it's going to want to go down its concentration gradient, which would be in towards the plasma. And this would be one way in which we can actually get rid of the waste that is accumulating. And other waste as well, not just CO2, but things like lactic acid and so on, or any kind of metabolites that might be accumulating from the cells themselves. All right. And as I progress through this lecture, you'll see that the barrier between the cells and the interstitial fluid, or the vasculature and the interstitial fluid, play a role in how fluid will shift between those compartments. So the capillary beds, they're actually a network of anywhere from like 10 to 100 capillaries typically, and they're supplied by a single arteriole. So for example, you have one arteriole that'll branch into 10 or up to 100 capillaries, and then that kind of coalesces into one single venule, and that's the typical structure of most of these capillaries. But another important thing is the capillaries themselves, they actually have these sphincters on the arterial side. And the sphincters are these little spherical sphincters that surround where the entrance of the arteriole into the capillary, and at those entrance points they can relax or constrict, and that will actually limit the amount of blood that goes into the capillaries. Okay, so when they contract, when a circular muscle contracts, it makes the diameter or the lumen smaller. So if it constricts or contracts, it'll limit blood going into that capillary. If it relaxes, it'll increase blood flow into that capillary. Now, importantly to understand is that the venous side does not have those sphincters, so it cannot regulate it. All right, but on the arterial side, it does. So take a look at this. What you have over here is our typical arteriole. All right, so we have over here, you can see the arteriole, and you see it's entering into here, and you see at each of these junction points along the arteriole there, that spherical structure there, all right, that is actually the capillary sphincter. And notice once we get to the venule side, you don't see any of the sphincters over here. They're all missing there. There are no sphincters on the venule side. And again, this allows us to limit flow into a region of the tissue if it's not required. So this is an important point, actually, physiologically speaking, is that organs throughout your body do not require a large blood supply at all times. A classic example would be as we're sitting here, or as you might be sitting here resting or sleeping, all right? If you're resting, your muscles are not working, not all the skeletal muscles are working that hard, and so you don't need a very large capillary blood supply during that time. So you might have constricted sphincters there to limit the amount of blood that's perfusing the skeletal muscle because it's not being utilized, and so that blood can be shunted somewhere else, so it can supply organs that might be working, like your brain, for instance. Now, on the other hand, if you were to say get up and exercise and do 100 jumping jacks right now, what's going to happen is you're going to increase blood flow to the muscles. And what happens is that's actually causing the arterioles, or the arterial sphincters there, the capillary sphincters I should say, to relax. And when they relax, it increases flow to those areas because now they're more metabolically active. So if you're exercising, your skeletal muscle is more metabolically active, which means it's building up more CO2 and lactic acid, but it's also demanding more oxygen and nutrients like glucose. So therefore, it's got to increase its blood flow. But at rest, it didn't need that, so it was able to constrict, which you can see those sphincters are closed in B there, so limiting its flow, all right, through the tissue there. Now, what's interesting is, that's the skeletal muscle example, but that could be any organ. The organ itself, some parts of the liver might be working more than other parts, and so some might be constricted and some might be relaxed. The brain is doing the same thing. Brain, if it's working, it doesn't mean the whole thing's working all at once. Parts of your brain might actually be more active than others, and so therefore there's an increase in blood flow to the metabolically active regions of your brain, and it's the capillaries can be more constricted to the ones that are not working as readily. So this helps us to distribute the blood flow as necessary for the metabolic need, so we can match it. So here I want to discuss the velocity of blood, the relative flow rate of blood, and what this graph over here is actually depicting is as we go from the arterial side to the venous side and we pass through the capillaries, the velocity of blood changes. And you can actually see it depicted here on the graph. So we'll take a look down here at the velocity of blood flow. You see it's highest in the aorta, and then that velocity over here starts to decrease in the arteries and really drops there in the arterioles and the capillaries. And what you'll notice is the velocity picks up again as we go towards the venous side. And this has to do with the cross-sectional area of the circulatory system. So for example, I could take a cross-section of the aorta here, okay, which is that single large artery exiting the heart, and I take the cross-sectional area. I take then the cross-sectional area here as it divides. Now that's the cross-sectional area of all the vessels in parallel that have divided there. Right? They're all, because when they divide and they create what we call parallel circuits. And so I take the cross-sectional area of all the ones that are in parallel. And that, of course, cross-sectional area would be greater than the aortas. And as I go further and further along, you see it becomes more and more branches create more and more parallel circuits. And if I take cross-sectional areas of them, and I basically add up all those cross-sectional areas of all the individual arteries and arterioles, the cross-sectional area becomes greater and greater and greater, and until we reach the capillaries, where the cross-sectional area is the greatest. What this means is that as blood goes from a small cross-sectional area to larger and larger cross-sectional area, because we have more and more parallel vessels, the velocity of blood flow slows down more and more and more. In fact, once it hits the capillaries, it's moving the slowest. Now this actually works out well for us because having the blood move the slowest through the capillaries allows us to do the exchange that's necessary with the tissues. So that actually helps us to optimize our exchange. You can think of it like this, as a river that's flowing into a lake. A river itself, as you can see it, with a very small cross-sectional area, moves very quickly. But when it hits a very large lake, the flow goes very slowly into the lake. Now if on the other end of the lake it forms back into, let's say, another river or another stream, you see the velocity of the flow picks up again. And that's effectively what's happening here with this with the circulatory system. High flow through the arteries until it reaches the capillaries where it slows down. It's like hitting a large lake. Then it recoalesces and the velocity picks up again. Okay, so here what I want to kind of point out, right, you'll notice in this cartoon the graphs are similar to the one that we just saw in the previous slide. So you have the arteries, the arterioles, capillaries, venules to the veins. And it's showing you, or it's depicting the same thing with the velocity here, where you can see that the velocity is highest in the the larger artery there. The arterioles, you start to see it drop, and the velocity decreases in the capillaries the most, and that coincides with an increase in the total cross-sectional area, as I mentioned in the previous slide. The reason I've added it here, though, is because in addition, it's also added blood pressure in millimeters of mercury. Now, blood pressure, I'm sure you guys have, what blood pressure is, or a normal blood pressure is an arterial blood pressure somewhere between 120 over 80. And we'll go into all sorts of details of blood pressure regulation and so on in the cardiovascular lectures to come. But at this point, I'm just trying to show you that the blood pressure, you see it holds pretty steady in the arteries, and then there's a drop in the arterioles. Blood pressure drops there. And then you'll notice, and this is important for the next few slides, is that the blood pressure drops across the capillaries as well. So at the start of the capillaries, you have maybe approximately 30 millimeters of mercury pressure there. And by the time you get to the end of the capillary on the venule side, the pressure may have dropped from 30 to about 10. All right, so there's a drop as we go across the capillary there towards the venule, and you'll notice the blood pressure continues to drop as we go from the venules to the veins back to the heart. Okay, so that's an important pattern to see right now. Now, the blood pressure itself, another term for that is called hydrostatic pressure, which I've written over here. So this is the hydrostatic pressure. This is the pressure that's exerted against the walls. It's a pushing pressure, which is why I put that in quotes there. this is pressure exerted outwardly against the vessel wall. Now imagine if I had pores in that vessel wall. If I push outward against it, some of the fluid is going to leak out. If they're small pores, it's going to be a little bit of fluid, and it's going to exclude larger solutes that won't be able to fit through those pores. And that's what capillaries are doing. They're basically exerting this outward force, and that's going to filter water out through those pores. Now that outward force is of course occurring all along the vasculature from the arteries to the veins, but the filtration only occurs in the capillaries because that's where the pores are located. Okay, and so the amount that filters depends on the amount of pores that there are, the size of the pores, and so on. And so that's a hydrostatic pressure. That's a pushing force that's trying to push fluid out. Now there's an opposing force called oncotic pressure. All right, this is also measured in millimeters of mercury. Now this is actually can be considered like a pulling force, if you will. This is really, in other words, this is like the osmotic pressure that we talked about in the previous lectures. Okay, but it's a special type of osmotic pressure that's exerted by the proteins in the vessels. The main protein being albumin. Albumin is the primary protein responsible for oncotic pressure in blood plasma. It makes up about 80% of it. All right, then there's globulins, which make up like another 20%. So these proteins are very numerous. So if you recall, the solute, the osmotic pressure of a solute depends on the fact that it's impermeable. All right, and it also depends on the number of solute molecules. So what this means is that the vessel, all right, doesn't allow proteins through those pores, as I've mentioned. So they stay inside the vessel, so it cannot get out into the interstitial space. And if you recall, the interstitial space and the vessel are all part of the extracellular fluid. But now I have a barrier between the interstitial and the vascular space that doesn't let the proteins through into the interstitial space, so it's effectively impermeable. So that means proteins get stuck inside the vessels. They're high in abundance, and so therefore do play a role in water shift or fluid shift between the interstitial space and the vascular space. Sodium chloride and glucose do not, even though those are our primary ECF osmolytes. That's because they play a role in fluid shifts between the cells and the interstitial space. Okay, I'm going to go into more detail of this in the slides to come, but suffice to say that these proteins, all right, are high in number and cannot get out of the vascular space or out of the vessel and into the interstitial space. So it becomes impermeable across that barrier, and so therefore it can keep or retain water inside the vessel. So we refer to that as like a pulling force. Since there's high solute protein concentration in the vessel, it's going to try to pull water back towards itself out of the interstitial space. All right, so that's why I refer to it as a pulling force. So we have two. We have hydrostatic pushing force, and we have the oncotic pulling force trying to keep the water from leaving. So these are the two opposing forces. Now these two opposing forces occur both in the vessel as well as in the interstitial space. There is a hydrostatic pressure there as well as an oncotic pressure. And so I'll describe both in some of the slides to come. Okay, so here what I want to just show you visually is the change in the pressure across the capillary, as I mentioned on the previous slide. There's a blood pressure drop from the arterial side to the venule side of the capillary. So let me show you what this looks like here. So what you can see over here is the arterial end of the capillary. Here's the venous end of the capillary and the direction of the blood flow. Okay, so here's our capillary, which you can see the red blood cells in there. All right, the red arrow is showing the hydrostatic pressure. Remember, that's the hydrostatic, that's the pushing pressure from inside the vessel. That's trying to make its way out, and you see it's greatest at the arterial end where the blood pressure would be the highest, around 30 millimeters of mercury. And the blood pressure drops as we move along the capillary until we get to the venule side of it, which is going to be about 10 millimeters of mercury on average. Okay, so there's a pressure drop, and it's depicted by the shrinking red arrows as we go from one side to the other. Now inside the blood vessel we also have the oncotic pressure carried out by albumin and some of the other proteins that are present in the blood that cannot cross that barrier from the vessel into the interstitial space. So it can't make it over to the interstitial space. So since it's left in the vascular space there, it can exert an osmotic effect, and we call that an oncotic pressure or a colloid osmotic pressure. And so that's going to be a pulling force inside the vessel, and you can see that depicted by the blue arrows. And you'll notice that the blue arrows stay the same size throughout because the oncotic pressure in the capillary doesn't change from one end of the capillary to the next. Right? That stays constant throughout. So although we have a pressure drop, the oncotic pressure stays the same throughout. And then what we do is we compare the relative forces to each other, the pushing and the pulling forces, to see whether we're going to actually push fluid out of the capillary or we're going to be, absorbing fluid into the capillary. So that's why we want to understand those forces. And as I mentioned on the previous slide, you'd have the same forces occurring in the interstitial fluid. I'll just write ISF over here, interstitial fluid. You still have some proteins that are there that are produced by the cells here. Okay, it's not proteins that came from the blood vessel. And you also have some fluid in the interstitial space there. So it's going to have its own hydrostatic forces and its own oncotic pressures. And so what we're going to do is compare all these forces to see how fluid is going to move. And this is all, the calculation for this is all based on his Starling forces. Okay, so I want to illustrate some of the points that I just made on the previous slide. So let me draw the capillary. All right, so this is the capillary here. That would be the lumen, would be inside here. I have blood moving in this direction. Okay, so that would make this side the arterial side, and this side over here the venule side of the capillary. Right now what I'm doing is I'm just drawing some oval shapes here just to be a marker of the endothelial cells. So these represent the endothelial cells. Draw another one there. All right, so that's the endothelial cells, and the black line would be the basal lamina. Okay. So the first point I want to make here is the difference between oncotic pressure, and osmotic pressure. So first off, I have to remind you from the first lecture, okay, that the intravascular space and the interstitial space make up the ECF, or the extracellular fluid. That's right, so that's the interstitial fluid and the intravascular fluid make up the extracellular space. Now I'm just going to draw some cells out here. So these are just some type of cell on either side as our capillary is running through the tissue. Okay. Now, as discussed earlier in this lecture, there are pores or openings between the cells, and in some cases actually pores or openings within the cells themselves. But I have little openings you can see right here. Okay, I've drawn a couple, that's why I draw them a little bit separate there. And so this allows the blood or the plasma in the capillary to be able to communicate with the interstitial tissue. And as discussed already, this is where diffusion is going to take place. Now, in terms of oncotic versus osmotic pressure, remember we always compare fluid shifts between the ECF and the ICF. The ICF, of course, would be the intracellular fluid located inside the cells themselves. So it's always the barrier, the plasma membrane barrier, that separates the ICF from the ECF. However, in the ECF, since we do have two parts, the interstitial fluid, which is the fluid that surrounds the cells itself, okay, and then we have the intravascular fluid. There is a barrier between the interstitium and, of course, the vasculature. All right, and so we can see that barrier here. I'm just going to darken the line right here, but this is our barrier, okay? The wall of the the vessels actually separates it from the interstitium. So we have another barrier, and anytime we have a barrier like that, we do have to account for if there's any differences in the way solutes can diffuse, because that can affect the way fluid will move, just like the way it would work across the plasma membrane. So let's take an example of that. One that we know that's an impermeable solute found in the extracellular fluid that we've discussed already in the previous lecture was sodium chloride. Sodium chloride is found throughout the ECF, which means it's both in the vasculature as well as in the interstitial space, with very little of it being in the intracellular space. Now, the reason it's found in both, and it's found in equal molar amounts, all right, so it's the same as osmotic pressure in the intravascular and the interstitial space, because sodium chloride can freely move between the vascular space and the interstitial space. So in this case, the barrier between the vessel wall and the interstitium, okay, is sodium chloride is freely permeable. So therefore, it's not going to act like a solute that's going to cause any effective water shifts between the vascular space and the interstitium. Again, because it can freely move, so therefore it can equilibrate via diffusion very readily. So it doesn't act like an effective osmole between at that barrier between the interstitium and the vasculature. Now, okay, so now if instead what I have is I'm just going to draw these little circles here, which are going to represent albumin, which is a small protein but found in great abundance in the blood. Okay, and in fact, these pores for most of the capillaries in our body are too small to allow albumin to diffuse into the interstitial space. So it is impermeable. If you want to look at it this way, it's impermeable to albumin. So albumin cannot get into the interstitial space. So it's a high concentration in the plasma, in the vasculature, but cannot get into that interstitial space. Therefore, it's impermeable at that barrier. So that means protein, since it is impermeable by its nature, can have an effect on the movement of water, all right? And that will, in effect, help to retain water, if you will, in the vascular space. The reason is because I have a high concentration of protein in the vascular space relative to the interstitial space, or the interstitial fluid over here. Now, there is protein in the interstitial fluid, just not as high a concentration as that in the vasculature with albumin and globulins and so on. And since it cannot permeate and get through into the interstitial space and equilibrate, it can have an effect on osmotic pressure. And so it actually does, and it can actually cause water, in some circumstances, to actually move back into, from the interstitium back into the vasculature. Now, that is osmotic pressure. It's the same principle that we talked about in the first lecture. The only difference here is that instead of calling it osmotic pressure, all right, this is osmotic pressure due to proteins, and so therefore it has a special name. So we're going to call it oncotic pressure. Another term for it is colloid osmotic pressure. It is really just to single out the fact that in the extracellular fluid, all right, the proteins exerts an osmotic pressure within the vasculature. The vasculature, which is separated from the interstitial fluid by that barrier that excludes the proteins. So therefore, it can have an effect on water movement or fluid shifts. Okay, so that is what we refer to as our oncotic pressure. It's the pressure, right, the osmotic pressure exerted by the proteins in our blood. The primary protein that causes an osmotic pressure is albumin, but we also have globulins, which can exert 10 to 20% of the effect as well. Now, what I want to also show you with this capillary here, I'm just going to erase this for a minute. All right. Now I want to talk about the pressures. So at the arterial end of the capillary, remember, so the blood is moving through the artery to the capillaries, all right? And the last artery goes through is the arterial and then into the capillary. Now, the pressure is higher on the arterial side than on the venous side. And we have what we call hydrostatic pressure, which I described in the previous slide. So the hydrostatic pressure, I'm going to use these numbers here because they're fairly average for many of the capillaries, would be 30 millimeters of mercury. All right, that's within the lumen of the capillary at the arterial end. And that is I'm going to put the capillary hydrostatic pressure. And on the venule side, as I showed you in the diagram on the previous slide, the pressure drops as we move across the capillary. On the venule side, the hydrostatic pressure is 10 millimeters of mercury. All right, so again, that's capillary hydrostatic pressure. So there's a drop from 30 millimeters of mercury to 10. And remember that hydrostatic pressure is a pushing type pressure against the wall of the capillary. So it's trying to force fluid out. And when it gets to these pores that are located in the capillaries, all right, that's going to cause fluid to move out. And of course, since the force is greater, the hydrostatic force is greater, all right, would have a greater pushing pressure at the arterial end than we would say towards the venule end. So I just drew a smaller arrow to indicate that. So it would drop as we go along the capillary, okay, towards the venule side, because we have a drop in that hydrostatic pressure. And again, the hydrostatic pressure represents our blood pressure, right? That's what we measure in our arteries. Now, another pressure inside the capillary is the oncotic pressure, which I just described, which is the osmotic pressure exerted by those proteins. And so the oncotic pressure, I'm going to put on O. All right, the oncotic pressure. Oh, let me rewrite this. Capillary osmotic pressure, all right, is about, on average, about 28 millimeters of mercury. And I drew it right there in the middle because, in fact, in many of the capillaries, all right, the capillary oncotic pressure doesn't change from the arterial side to the venule side. So that stays constant from one end to the other. So that is throughout. All right. So those are the pressures. So what these are two opposing pressures essentially. You have the hydrostatic pressure, which is a pushing pressure, which is going to try to force or filter fluid out of the capillary, which I drew with those blue arrows. But now you're going to have an oncotic pressure, which is caused by the proteins trying to pull water back in or draw water again towards the higher solute concentration, which in this case would be the proteins. And so that's going to have a fairly constant force throughout, which is why I drew the arrows all about approximately the same size. It's going to try to draw water into that capillary, while the hydrostatic pressure is going to try to filter or force it out of the capillary. So we have two opposing forces. Okay. Now you can see from this that if we were to take a look at the arterial end, if there's a 30 millimeters of mercury pushing force and only a 28 pulling force, that we would probably filter more at the arterial end. And by the time we get to the venule end, we only have a 10 millimeter mercury pushing force and a 28 millimeter mercury pulling force. We probably absorb at this end. So we'd probably absorb fluid here on this end, and we would filter on the arterial side. And that's actually what happens, but this is not the entire story because we do have to account for what the pressures are in the interstitial fluid, because this interstitial fluid that surrounds these green cells that I drew here also exert a force. So let me pick another different color here. I'll use purple. So in the interstitial fluid, again, that's the fluid out here surrounding the cells, interstitial fluid. There's also a hydrostatic and there is also an oncotic pressure. The oncotic pressure is exerted by proteins that reside in the interstitial fluid that have been produced by the cells. These have not been filtered out of the blood. And on average, that's going to be 8 millimeters of mercury. All right, we call this the interstitial oncotic pressure. All right, and in addition to the that oncotic pressure, we also have a hydrostatic pressure, which again is typically like a pushing force. But in the interstitial fluid, on average, that force, that hydrostatic force in the interstitial fluid, is actually very low. Most cases, the hydrostatic pressure is actually 0. It could be 0 millimeters of mercury to maybe -3 millimeters of mercury. And this again, this would be interstitial hydrostatic pressure. So 0 is essentially no pushing force whatsoever. There's no acting hydrostatic force. What does negative mean in terms of a hydrostatic force? Well, negative force would actually imply that there's a vacuum. That instead of a pushing force, we really have a pulling force now because it's acting like a vacuum. Okay, and there are lymphatics that are located in the interstitium there that actually suck up excess fluid, which I'll discuss in a little while, and that can actually create a vacuum-like effect. So in fact, the hydrostatic pressure oftentimes is even negative. Now I drew them over here and not towards the arterial or venule side because the interstitial oncotic pressure and hydrostatic pressure, they're constant throughout the length. So those are forces that are acting throughout the whole length there. All right. So the question then becomes, well, how is fluid going to move? And it really comes down to a simple arithmetic, looking at the numbers. Okay. We're sort of adding up what these, looking at what the hydrostatic pressures are. Let's look at what the oncotic pressures are. Let's tally it up, and then let's see what the net filtration pressure is. Something we refer to as an NFP, net filtration pressure. All right, and what that really equals, all right, it's simply put, is the capillary hydrostatic pressure. So we're going to look at the hydrostatic pressures first, minus the interstitial hydrostatic pressure. Oops, that's a minus. All right, minus the capillary osmotic pressure, minus the interstitial osmotic pressure. And so we can tally that right now and kind of see what that looks like. So for instance, our capillary hydrostatic pressure here is 30 millimeters of mercury, minus the interstitial hydrostatic pressure, which we'll go with -3 since many of the interstitial fluids is roughly negative. So we'll say negative, so minus some -3 is actually +3, right? And you take the capillary oncotic pressure, which is 28. And by the way, I'm using the numbers, obviously the hydrostatic pressure I'm using from the arterial side. So right now I'm calculating the net filtration pressure on the arterial side of the capillary. The arterial side of the capillary. And so this is, let's go back to the oncotic pressure. So that's 28 minus the interstitial oncotic pressure of 8. And so that's 28 minus 8, right? So we have 33 minus 20, which comes out to 13 millimeters of mercury. What does that actually mean? How do we interpret that? Well, a positive 13 millimeters of mercury means that there is a net positive filtration pressure out of the capillary of 13 millimeters of mercury. So we are filtering at the arterial end with a pressure of about 13 millimeters of mercury. Right? We're accounting for both the pushing and pulling forces, both in the capillary as well as in the interstitial space. Now that's the arterial end. So we have 13, that's the net filtration pressure is 13 on the arterial end. If you look at the venule end, we're going to do the same calculation, but really the only thing we're changing, if it's going to be the venule side, is we have to change that capillary hydrostatic pressure because it's no longer going to be 30. Instead, that's going to be 10. So I'm going to have to erase some more things here. Okay. So right, so the 10 again comes from over here. We're looking here at the capillary hydrostatic pressure. All right, minus the -3, which is +3. That's going to be 13 minus, and again, the oncotic pressure in the capillary did not change, and the oncotic pressure in the interstitial space did not change. So that's a 28 minus the 8 again, so that's 20. So 13 minus 20 comes out to a -7 millimeters of mercury. So on the venule side, the net filtration pressure is -7 millimeters of mercury. We interpret a negative as an absorption. So really, since there's a negative pressure there, we're actually absorbing fluid on the venule side of the capillary. So what you're seeing occurring is what we just predicted earlier, which is that you're getting filtration at the arterial end, and you're getting absorption of fluid on the venule side. And so based on the calculations that I just went through with you, I use the same numbers, and you can see that all right here. Okay, so you can see that on the capillary side, on the arterial side of the capillary, excuse me, that was a 30 millimeters of mercury hydrostatic pressure out, right? And then the interstitial space was -3. Instead of saying -3, it just said positive out of the interstitial space. Okay, because it's acting like a vacuum, so they're working together. So it's effectively pulling out since it's a negative hydrostatic pressure, and so that comes out to a net hydrostatic pressure of 33 millimeters of mercury. Now, if we have to look at the oncotic, which is the opposing forces, okay, inside the vessel itself, all right, we have an oncotic pressure of 28, so that's trying to pull fluid into the capillary, and then -8 would be the oncotic pressure in the interstitial fluid, which is trying to pull the fluid into the interstitial space. So they are opposing forces, and so that gives you a net oncotic pressure of 20 millimeters of mercury. So then we compare the hydrostatic versus the oncotic, that pushing versus pulling forces, and it looks like the pushing force wins out with 13, right? So you can see, you can calculate the same thing on the venous side and do it for yourself again and just kind of repeat it for practice sake, but that's the general idea. In terms of filtration and reabsorption, right, this is kind of already summarizing everything I just talked about. Where primarily in most capillaries, most continuous capillaries in the body, you're going to have filtration occurring at the arterial end when the pressures are higher, and you're going to have reabsorption on the venule side. Now, there are conditions where that can change, and I'll go through some examples of that later. And you also have different types of specialized capillaries that we'll get into more details about those specialized capillaries when we talk about those specific organs in those lectures. All right. But here, what I want to point out is you can see in the red box the net filtration pressure of the arterial end of the capillary, and you'll notice there's the hydrostatic pressure of the capillary, hydrostatic pressure of the interstitial space, the oncotic pressure of the capillary, oncotic pressure of the interstitial space. So again, it's the calculation that I just did for you guys. The only difference which you might note right now is you'll notice that there's some modifiers here. Right, you see this KFC and the sigma sign here. So these are modifying the equation. So let me explain that. So the KFC, all right, the filtration coefficient, this is basically asking how easily does fluid move across the capillary when the force is applied. It's really just, it's a modifier for the equation to account for the permeability of different capillaries. Not all capillaries are created equally, so even if we're talking about continuous capillaries, they can be different. They have different permeabilities based on the number of pores that they have, the surface area of those capillaries. So this can all change the permeability of it, and so we can account for that by, giving this this coefficient. All right, where you can see the descriptor over here to the right. If it's very small, for example, like 0.01 milliliters per minute per millimeter of mercury. Right, so that's how much fluid is going to move every minute for a single millimeter of mercury. You see, it's very, very little when it's 0.01, so a very small filtration coefficient. That might coincide with something like, say, the blood-brain barrier, okay, where the permeability is very little. On the other hand, 1, let's say 1 is our standard here. That might be most of our continuous capillaries, might show that. However, it could be 100 or 1,000 times, and that just means the permeability is increasing several fold each time. So, for example, in the sinusoidal capillaries, which have very large intercellular clefts, you could have, very large permeability, and so therefore we have to account for that in the equation, and so we can modify our equation with the coefficient, all right, to indicate that. Because obviously if I apply the same pressure to one that's very permeable versus the same pressure to one that's less permeable, I'm going to be moving more solutes in the very permeable capillary. Now the sigma, which I showed you in the previous slide, the sigma is a modifier for the oncotic pressure. All right, what this really means is this is taking into account particular solutes, okay, and how easily they can cross the capillary wall. It indicates the size of the pore. The larger the size of the pore, the more likely the solute's going to be able to move out of the capillary and into the interstitial space. And usually we do this as from 0 to 1. All right, so that's the scale. So if it's 0, for example, what we say is that 0 means there is no reflection. So this thing can completely move through the pore without any problem at all. So for example, if let's say we're talking about albumin and we said, well, in this capillary, the sigma is 0 for albumin. What that means is that albumin can move freely through that pore. Okay, it can move freely. It doesn't reflect back into the capillary at all. And that's something you might see, for example, in a sinusoidal capillary. Versus say, a scale, or excuse me, a sigma of 1, that means it's completely reflected. That means it can't get out of the pore at all. It does not move through. So albumin, for example, has essentially close to a sigma of 1 when we talk about continuous capillaries. The reason it modifies the oncotic pressures is because, for example, let's say we're going through a sinusoidal capillary. Protein can easily move out of the capillary into the interstitial space because the pores are very large, right? So in that case, the oncotic pressure doesn't take, we can't take it into account anymore because it can move across the barrier of the vessel wall into the interstitial space. And it's permeable now, so therefore it doesn't have an oncotic effect. So if it's 0, 0 multiplied by the oncotic pressures, right? So it's just going to come out to 0. We're just worried about the hydrostatic pressures in that equation then. On the other hand, if it's in the continuous, it's close to 1, right? And then there will be some capillaries where it might be somewhere in between. So again, really it's kind of important to understand how they're modifying, what they're modifying, but I won't expect you guys, or I won't give you guys questions based on calculating, the filtration coefficients, but we should understand, what they represent. Okay, so the hydrostatic pressure, that pushing force, right? It's exerting that force and it's pushing fluid out. You got the oncotic, which is pulling it back in. And as I mentioned before, that hydrostatic pressure represents blood pressure, right? That's what our blood pressure actually is, okay? Now, in general, if fluid gets filtered out of the capillaries will reabsorb, as we saw on the venule side, it will reabsorb about 85% of the fluid that's filtered. So what you'll notice is that 15% of the fluid is not reabsorbed, okay? Now the problem with that is I can't leave behind 15% every single time blood is moving, at all times when blood is moving through there. If it's always leaving behind 15%, it's going to accumulate, and I'm going to have edema. But that's not the case. That's not under normal physiological circumstances, and that's not what happens. Instead, there's what we have, the lymphatic system, and the lymphatic system is connected to the circulatory system. We'll get into more detail on that later, of course, but the lymphatic system basically acts like a vacuum. It has open ends that will actually vacuum out the excess fluid or any kind of protein or solute that may not have or should not be there, and it will vacuum that out, move it back into the circulatory system or back into the blood. Okay, and so that's how we can get rid of anything that's left behind. So it plays a large role in maintaining our fluid balance. So for example, clinically speaking, if you had a problem with the lymphatics, or if there was obstruction of the lymphatics, or let's say you had a tumor that was obstructing the lymphatics, you could see massive edema accumulating there because the lymphatics are obstructed and it can't get rid of the excess fluid, and so you can start to see very large accumulations of fluid. So speaking of edema, right? So now we got to talk about really kind of the clinical aspects of all this fluid shift. So we talked about, you know, the physiology, which is how fluid is being filtered and reabsorbed, and how we have diffusion occurring during that process, right? And transport, and that the blood is moving slowly, so it optimizes our timing and so on. But when it comes to the actual pathology, or what you might see in patients when there's a change in fluid status, right? Either fluid loss or fluid accumulation, what can that present like, right? So for example, in fluid loss, you have dehydration. You could have a hemorrhage, right, where you're actually losing whole blood. And I'm going to show you on the next slide, there's a process called autotransfusion. So how does our body compensate for fluid loss? How does it shift fluids in our body to help maintain the vascular space? Because if we continue to lose fluids, all right, we're going to reach a threshold where the body can't compensate anymore. And that's when you start to see real disease and very sick patients. But up to that threshold, you know, the body is able to compensate, and I will show you graphically how that's done. Then you have the flip side, which is fluid accumulation, or, you know, the general term for it would be edema, right? Swelling. So we've probably all seen that at some point, right? If we're standing for too long, you know, your feet and your ankles can swell, for example. Some people may have something called ascites. So you're going to see this in your patients. So over here in this picture here, that's not pregnancy, okay? That's ascites. And so this could be someone who has, say, liver failure, and the liver failure is causing fluid to back up and increasing pressures, and it's causing an increase in accumulation of fluid in the capillaries there, and it's leaking fluid into the abdominal cavity, causing what we call ascites, which is really just, you know, a form of local edema that's happening there in the belly. You could also have what we call generalized edema, which is basically edema everywhere. So that's the difference between, you know, edema types really, is you have generalized and local. You know, local can be you have an injury of your ankle, if you twisted it, you see swelling there, versus generalized where you see it everywhere. So in the picture of the child here, you see this is what we call anasarca, and it's a low, you know, protein deficiency, and usually due to diet in this particular country. And you can see just the swelling everywhere. And again, that lack of protein means a lack of oncotic pressure, so it can't keep the fluid in the vessels, and so it can accumulate in the interstitial spaces, and that's what you're seeing there. And not only is it happening, which, you know, physically where you can see it, but this would also be occurring in the organs as well, and can cause damage to the heart, for instance. So other, you know, causes of edema would be things like heart failure, cirrhosis, which is, you know, disease of the liver, inflammation, again, like I said, like an ankle sprain or something like that, and something else called the nephrotic syndrome, which is actually a disease of the kidney, where the kidney, it filters blood, right, and it does this job that's, you know, when it's filtering, of redistributing electrolytes and solutes to their appropriate places and the appropriate quantities, and as long as it's functioning appropriately, everything is, you know, everything is there to maintain homeostasis. But in some cases, if the kidney is damaged, it might filter proteins, which it's not supposed to, through its capillary, and the protein can end up in the urine. So proteins like albumin. So in that case, if you lose the albumin, you can become, you know, edematous, simply because you have no longer have the ability to retain water or fluid inside your vascular space. So these are just some examples. There are many other examples of things that can cause fluid shifts. So what I'd like to do now is kind of take you through graphically representation of capillary fluid shifts and kind of address some of these issues. So I like this graph because it can actually demonstrate fluid loss or fluid accumulation at the level of the capillary, based on the various changes that I spoke about on the previous slide, having to do with dehydration, for example, in fluid loss, or say loss of protein, causing drops in oncotic pressures and so on. And so I can actually represent that on this graph. But what I want to show you on this slide first is how to sort of read this graph first, and then I'll show you, I'll draw some graphs to kind of depict some of the various situations. So to start, you'll notice here that on either axis I have millimeters of mercury, so that's just the pressures, okay? And on the axis here is the capillary, which is actually, this is the arterial end of the capillary, and this side represents the venule end of the capillary. So again, blood is flowing in this direction towards the venule. Okay? So let's take a look at the arterial end. Now again, this is really just graphically representing what I showed you with the net filtration pressure calculations earlier. So let's use those same numbers. All right. So for example, if we look at the hydrostatic pressures, remember it was about 30 millimeters of mercury on the arterial side in the capillary. And it was about -3 millimeters of mercury in the interstitial space, and so it was approximately 33. Give or take, I didn't draw it exactly at 33, but that's why I drew the net hydrostatic pressure on the arterial end to be approximately around 30. And you'll notice that it drops as we go along the capillary, because at the venule side we calculated that too. That was approximately 10 millimeters of mercury inside the capillary, and it was about, again, still -3 in the interstitial space. So that would have been about 13, which is an approximation right there. It's close to about 10 right there. So you can see that the drop in pressure went from 30 on the arterial side to 10 on the venule side. So that line, that blue line there, represents the net hydrostatic pressure throughout the capillary. Now, the orange line here, you'll notice that it starts out with a net oncotic pressure. So it represents the net oncotic pressure. On the arterial side, it's about the average is 20. How did I get that? Well, if you recall that inside the capillary, that was the oncotic pressure was 28 millimeters of mercury. In the interstitial space, the oncotic pressure was 8 millimeters of mercury. So I subtracted one from the other since they're opposing forces, right? The proteins pulling from the interstitial space and the proteins pulling into the capillary space or into the vascular space. They're opposing forces. So I subtracted one from the other, and you get 20, right? 28 minus 20. And if you recall, those oncotic pressures, both in the interstitial space and the capillary space, don't change throughout the length of the capillary. So that's why you see a nice straight, again, linear, and the pressure doesn't change, where it goes from 20 millimeters of mercury on the arterial side and 20 on the venule side. So that would be the net oncotic pressure throughout the capillary during that time. So this is representing those net pressures. Now, you'll also notice, all right, that here if we look at the net hydrostatic pressure on the arterial side, that at all points here and here and here, all along the net hydrostatic pressure line, are all greater pressures than the net oncotic pressure. Meaning that the pushing pressure, quote unquote, all right, is greater than the oncotic pressure at all those points until we reach this point right here, where they're going to be equal to one another and there will be no net movement. So what this triangle actually represents, I'm going to shade it in, this triangle represents right here is filtration, which occurs, as you can see, for almost more than half of the capillary's length. We're filtering because the hydrostatic or pushing force is greater than the absorbing or pulling force of the oncotic pressure. However, once we cross that equilibrium point, you'll also notice now this triangle, where now each of the points on the net oncotic pressure are actually greater than the hydrostatic points, meaning that the pulling force is now greater than the pushing force in the capillary. So therefore, that triangle represents reabsorption, or our net reabsorption. And so you see how it's a good visual of how the absorption or the filtration changes to reabsorption towards the end of the capillary. Again, this represents graphically the Starling mechanism, which I mentioned on the earlier slides. Okay, so here I'm going to redraw that graph as best I can, since I don't have a grid here. All right, so bear with me. But this is the arterial end, and this is the venule side. Okay. Again, remember, just the blood is flowing in that direction. And then we have pressure on each side here in millimeters of mercury. All right, so this will be 0, 10, 20, And again, here I'm just eyeballing it against, I don't have graph paper, so bear with me. Okay, so I'm going to keep the same color code as we had on the previous slide. So our net oncotic pressure, if this were normal, would be straight across, as you recall. And my net hydrostatic would go from this point to this point here as it drops from the arterial side to the venual side, and that is not straight at all. Let me see if I can do better than that. That's when I wish I had a ruler. Now it's a little better, but you get the idea here. Is again, this is our normal, and I showed you how we had our net filtration, which is occurring here, and then reabsorption occurring towards the venual side right there. So now, let's take an example of a clinical case where we have volume loss. Let's say we have dehydration, and we'll say it's due to excessive sweating. And if you recall from the first lecture, with sweating, we lose more water than we do solute. All right. So let's see what that would look like. Now, how does that affect capillary exchange? We have a fluid loss here due to dehydration, due to excessive sweating. So initially, what's going to happen if I lose water in excess of solute? That means I'm increasing the concentration in my blood, right? Because I'm losing more water than solute, so the ratio has been changed. So I'm actually going to increase the concentration. So in effect, what's going to happen to my oncotic pressure? My oncotic pressure is going to go up. So I drew those little arrows to indicate that that level would actually rise, because now the proteins in my blood are more concentrated than they were before because I've lost more water, right? So that concentration actually is going to go up. Now I'm not working with specific numbers, I'm just kind of showing you generally what's going to occur. So I'm just going to draw, another dotted line here to represent, let's say, maybe our new level over here. All right, so it's risen. So from 20 millimeters of mercury, maybe the net now is 25 or 30 or what have you, okay? Now what happens, or does anything happen to my net hydrostatic pressure represented by the blue line? Well, sure, if I'm dehydrated and I have volume loss from my vasculature, my ECF, well then, yeah, I can also have a decrease. I'll draw a downward arrow, a decrease in my blood pressure, because that's what happens when you're dehydrated. Your blood pressure can also go down, correct? And that's what hydrostatic pressure represents. And so I'm just going to draw it again, another line. I'll just pick some numbers that are lower here. Again, don't hold me to the exact numbers here. I'm not doing exact calculations. And so let me erase the original lines here, the normals. And I want you to take a look at the difference now in those triangles. So for example, my filtration is very small because my oncotic pressure has increased, or my pulling force, and my pushing force, or the hydrostatic force, has decreased. And look what happened now to this triangle over here, my absorption. Well, since my hydrostatic pressure dropped and my oncotic pressure increased, I now do more absorbing than I do filtering. And this, if you think about it, is actually a very cool way that the body can adapt to dehydration and maintain fluid inside the vascular space. So I don't want to filter out too much now if I'm already depleted, and I can actually reabsorb more than I filter, which is a very neat mechanism during a state of dehydration, for example. And we refer to this as auto transfusion, which I mentioned on the previous slides. Auto transfusion just means you're self-transfusing, right? Instead of us actually giving an IV bag to help replenish the fluids, which maybe this patient might need ultimately, but to help maintain homeostasis, the body automatically tries to reabsorb more at the capillaries than it does filter, and this helps to maintain fluid in the vascular space so we don't have vascular collapse. So it's a pretty neat mechanism. Let's try another example of fluid loss, another example of autotransfusion. I'm going to redraw my lines here so we can see and compare. And that's the problem with drawing dotted lines, especially when you're racing with the computer. So bear with me a second. Okay. So I'm just going to redraw our set point, our normals. So we're back to, let's say, our normal point right now. All right. And this time we're going to have fluid loss, but it's going to be in the form of an injury, let's say, as the person has a hemorrhage. All right, so this person hemorrhaged. So let's say they've had, some sort of an accident now and they're bleeding. And when someone hemorrhages, all right, the key here to a hemorrhage is that when they're losing blood, they're losing whole blood. That means they're losing the plasma, which has the water in it, and all the solutes that are in it, and they're losing it in equal amounts to each other. Unlike with sweating, where sweating we lost water, but we left the solutes behind so the blood could concentrate. But in this case, that's not what's happening. Okay, in this case, we're losing whole blood, so it's not concentrating at all. All right, I'm not losing more water than solute or solute than water. I'm losing both equally. So the concentration doesn't change. So in this case, there's no change to my oncotic pressure per se. Okay? However, what can happen is since I'm losing blood, I can have a drop, and depending on how much blood I've lost, I can have a fairly significant drop in my blood pressure, or in other words, my hydrostatic pressure. So I would expect I'm going to draw the down arrows here. So let's draw that dotted line there. So I've had a drop there. I'm going to erase my normals here. Oops, I didn't mean to erase my oncotic pressure normal. I'll draw that back in. And so what you can see here is it's effectively the same thing we saw before, only now, we see there's an increase in the absorption like we saw before, but only now, instead of the oncotic pressure changing, it didn't change really, or it would change very minimally. But it did have a bigger impact on the hydrostatic pressure, and it still ultimately led to the same effect, which is an autotransfusion, where we end up with a lot more absorption there. And again, it's a way for the body to try to maintain homeostasis and maintain fluid in that vascular space. Later on, we'll talk more about when the volume exceeds the body's ability to actually be able to compensate effectively, right? And that's of course why we intervene at those times. All right, so I'm going to give another example here. I'm just resetting my normals again. So I'm back to my normal working position. Now in this case, what I want to show is if we have an accumulation of fluid, which can often represent itself as edema, as I described in the earlier slides. So let's take an example. So one clinical example is I have edema. Now there are a multitude of things that can cause edema, one of which is referred to as nephrotic syndrome. Something you will learn more about in detail about later on, but suffices to say at this point that it's a kidney disorder wherein you lose proteins in your kidney. You should not end up having any protein in your urine. If you have protein in your urine, that's a sign of kidney damage or kidney disease. And so in this case, the kidney is getting filtered out and ending up in the urine where it shouldn't be. That means that the protein that's ending up in your urine is proteins like albumin. And as I've talked earlier on in this lecture, albumin is one of the primary proteins to maintain oncotic pressure, which helps to hold the fluid inside the vascular space. A loss of the albumin results in extravasation of fluid out of the vascular space because there's really no oncotic pressure to help pull it back in. So that becomes very dangerous, and so you can end up with what we call generalized edema from that, since that would affect essentially all parts of your body with that drop in the protein. And so as you would expect from that, you'd see a drop in the oncotic pressure, because if I'm losing albumin, I can't maintain the pressure. Right? I'm losing the solute that's responsible for maintaining or pulling water into the vascular space there. So it drops. All right. Now, again, the hydrostatic pressure over time would also drop too, because as the fluid leaves with the protein, you might also see that that drop as well, but at least initially. So we'll just theorize how it would look initially. You would see that there would be a drop here, and we'll draw the new line right here. All right, a drop in the oncotic pressure. And we'll leave the hydrostatic pressure alone for now, saying that maybe this is earlier in the disease when we're just losing protein, but the volume is still fairly steady. All right. Keeping in mind that, of course, as we lose those proteins and fluid moves out of the vascular space, of course, the hydrostatic pressure could drop too. But the initial thing, the initial insult, you would see something that looks like this. And now you can see this very large filtration triangle. So that kind of works against us, which is why the fluid would also move out even more so, because not only are we losing the proteins responsible for holding the water inside the vascular space, but now the filtration is much greater than the oncotic pressure. And so more fluid is going to get filtered out through a greater range of the capillary itself, with very little, if any, reabsorption occurring at all. This leads to, of course, edema, because now you have accumulation of large amounts of fluid in the interstitial space. And that can happen throughout the body, including into the organs themselves, and they can swell up with this excess fluid. And you could also lose, ultimately, like as I mentioned before, volume from the vascular space as well, which can lead to things like vascular collapse. All right, I want to do just a couple more examples to help kind of solidify, or at least reinforce our understanding. So I'm just drawing our baseline again. And we're still keeping with the edema, so it's fluid accumulation. Only now the fluid accumulation, all right, let's say is due to a different condition. Let's say we'll take heart failure. I'm just going to put HF here for heart failure. At this point in time, I don't expect you to know all the ins and outs of the pathophysiology of heart failure. So I'm going to give you the information that's sort of necessary. But we'll describe that and see how that fits into this picture here. So again, heart failure, the heart is a pump, it's a muscular pump, and it moves blood into the arterial system, right? And then the blood returns via the venous vasculature, all right? And that's how we kind of have what we call our circulation, right? Now, what happens is this: if I go into heart failure, that means the pump is not working correctly. And remember, the pump is generating that pressure or that force that's needed to drive blood through the entire vasculature. So if it's not working, that means that the pressure in the arterial system would go down, which would make sense, right? Because it's pumping, and if the pump fails, then the arterial pressure might actually decrease. However, there are mechanisms or fail-safes that help to kind of maintain that. And so we'll assume for now that we're not in complete decompensating heart failure, and that we do have some mechanisms that are helping to maintain the arterial blood pressure. And so that'll actually maintain the hydrostatic pressure at around 30. Okay, so we'll assume for now that's the case. So it's able to do that on the arterial side. However, if the pump is not working effectively, it means it's not moving blood through the heart very well. So it's not getting blood from the venous side into the arterial side, and so that means the blood starts to back up in the venous side. Now, as blood starts to back up on the venous side, that can start to pool and build up pressures in the venous system. And the venous system has very low pressures to begin with, and it has very thin walls and doesn't deal well with a lot of pressure. And that pressure can actually move backwards, and it can move back towards the capillary. And as I said earlier, there are no sphincters that separate the venules from the capillary. So the venules can't constrict sphincters and prevent backup of fluid into the capillaries. So what ends up happening is pressure builds up in the venous side. It can actually translate and back up all the way into the capillaries themselves and cause an increase on the venule side. It can cause an increase in the hydrostatic pressure on the venous side, while the arterial side is maintained normally due to our compensatory mechanisms, which of course we'll get to later on. But just as understand that that's the case right now. So, for example, we maintain our arterial side due to the compensation, but now I'm going to draw a little dotted line here to represent this point here. Let's say it changed. Our venous pressure went from 10 up to say 15 or 16 or 17 or whatever that point is right there. All right, so we had an increase on the hydrostatic pressure on the venous side only because the venous return has been decreased, and so venous pressure builds up and it backs up to the capillary. And so that affects that pressures there, or those pressures there. I don't necessarily have any change in my oncotic pressure at this point because I haven't changed the ratio of water to solute at this point. So the albumin concentration hasn't really changed. So really the only effect that's changed was an increase in the venous side, and that results in this effect. And so if I erase the original hydrostatic line there, what you're left with is very little reabsorption in that little triangle there and much more filtration. So it causes, again, fluid to move out of the capillary, filter out, and into the interstitial space and causes edema. Now, this can cause local edema as it starts to just initially back up and maybe affect organs, maybe even something like the liver, or it can affect gravity-dependent structures in your body like your legs and your feet. So you might see ankles swelling up and so on. Okay, so let's try one more example and we'll stop there. Okay, so now let me draw back my normal. I apologize for the glitch there. I just had to redraw the graph quickly. I just kind of put us back to our reset point there. You can see now our orange line again represents that oncotic pressure, the blue line the hydrostatic pressure across the capillary. And in this last example, I just want to give you an example of a mixed cause for edema, one with the term called cirrhosis of the liver. Now, cirrhosis of the liver affects it in multiple ways, depending on how severe it is. It can affect its functionality, or it could also affect it anatomically. And what I mean by that is this. So if I have, let's say, moderate to severe cirrhosis where it causes a lot of scarring of the liver, that can cause constriction of the blood moving through the liver. And the liver receives a lot of venous blood moving through it, so it can help to detoxify. For example, blood leaving from the digestive tract travels through the liver. Okay. Now, that would be part of its function, would be to detoxify, but if it's constricted physically or anatomically and it's constricting the blood vessels, again physically, that can obstruct flow and obstruct that venous flow that goes through it. And if it does that, that can back up pressures on the venous side, similar to what we saw in the example with heart failure. So the venous pressures could increase. All right, the venous hydrostatic pressures could increase from that backed up pressure, and we'd end up with a hydrostatic line. I'm going to draw that line, dotted line over to here, where we get an increase in the venous side hydrostatic pressure. Like the arterial side would not necessarily be affected. Now, and for similar reasons that I discussed with the heart failure example. Now, as far as the oncotic pressure, would that change? Well, again, it would depend on the functionality of the liver. One of the other jobs of the liver besides detoxification is that it's also there to produce proteins. And it produces a whole host of different proteins, and one of the main ones, which we've been talking about, is albumin. And as you know, albumin provides the majority of the oncotic pressure in the vascular space there. So if it's not producing that protein, we're going to lose that oncotic pressure, and so therefore the oncotic pressure would decrease. If I redraw that line there, okay, you can see the oncotic pressure decreased and the venous hydrostatic, or the venual side of the capillary hydrostatic pressure increased. And the net result of that, okay, is to actually cause, I'll kind of shade in the triangle here. Is to cause more filtration. So what happens is now in that those capillaries that are downstream of the liver, or should maybe say upstream of the liver, excuse me. When the liver becomes scarred and blocks the flow through it, that venous blood going through the liver is now backed up to the capillaries, which causes them to actually increase the amount they're filtering. And if you want to add on to the fact that it's also not producing the albumin, so the oncotic pressure is dropped, you can have the result being filtration, more filtration, and thus edema occurring. Now, if the liver were just scarred and it was just physically blocking venous flow, but it still maintained its functionality, so our oncotic pressure didn't change much, well, then you might only end up with maybe some local edema as it backs up blood into the local area. And if it's, in some cases that can cause things like ascites, which is really the fluid accumulating in the abdominal cavity there. Or if we're really losing a lot of albumin, that could become more generalized edema everywhere, because that would affect most of the capillaries there. Okay, and so finish up this lecture here. What I want you guys to try is I want you to read through this case, and there are questions that follow. And so these are the questions that go along with the case, and I want you guys to try your best and see what you guys can do with this. This is hopefully to kind of bring the first two lectures together and put it into some sort of clinical context for you guys. When I see you guys in Zoom, we'll go through this and I'll answer any questions you guys have regarding the lecture. Good luck.